think about your very first baby picture, right? The egg from mom, sperm from dad, come together to make you. Well, that process actually happens on a very microscopic level. So you can see here, I've given you a little reference that the egg is about 0.0047 inches and the sperm much, much smaller than that. But really, when you started, you were a tiny, tiny speck, a dot. Well, you're not a speck today, right? If you're watching this video, you're likely much bigger than when you started as this small little zygote. How did that happen? How did we get to be these big things from such a small thing that started so many years ago? That's what we'll look at in this video. Let's get started. Okay, from zygote to adult, how did we get to where we are today? Well, we got to be large adults because our cells divided. Cell division has a few purposes. One is reproduction, one is growth, one is repair and renew. So let me give you bacteria as an example. So bacteria, as you see here, for them to multiply, they actually divide, right? We say that biology is the only subject where multiplication and division are the same thing, right? To multiply, bacteria divide. So there's one bacteria, they double their DNA, then it divides into two. So one bacteria became two. Now we have two different bacteria. Actually, they're not so different, right? Most bacteria are just genetic clones of each other, unless there's been some mutation. So bacteria undergo this process called binary fission, where one just divides into two. Now our cells undergo a similar procedure, which we call mitosis. We'll get to that in a moment. So our cells divide, but the cells also have a life cycle. So the cell cycle is just the life of a cell from its origin to when it divides into two new cells. So think about back to that baby, right? Here's a mouse zygote development, right? We started as one, then we became two cells. Those divided into four. Over time, these cells will multiply and then start to differentiate. Here's another example of a ciliate that's undergoing cytokinesis, this final step where one divided into two. So you can see this used to be one, and then it has finally divided into two different ciliates, okay? So how do we go from zygote to adult? We divide to multiply. All right, let's talk about the difference between DNA and chromosomes. If someone were to say, oh, that's in your DNA, that's in your chromosomes, most people just think, oh, that's the same thing. Well, it pretty much is, but there are different levels, and that's what we need to learn right now. So, DNA, chromosomes, chromatin, how does it all work? So let's start. All right, we've got a cell. Here's our big cell. In the cell's the nucleus. We know that's where all this stuff hangs out, the DNA, chromosomes, chromatin. Everything, if you break it down to its level, well, let's start with DNA. You may remember DNA is a double helix, right? You see the one line, the two, in this helical pattern. This DNA wraps around these histone proteins. So the DNA gets wrapped around these proteins, then these things start to condense. This, when it's coiled, is now called chromatin. So, we got our first definition. Chromatin is just DNA wrapped around these histone proteins. But notice this is not yet coiled into chromosomes. So the DNA wraps around the histones to make chromatin, which is now going to condense even more into a chromosome that we find in our nucleus. Now, chromosomes we typically only see in our cells when mitosis is happening. Other times, the DNA is just hanging out as chromatin. All right, so what's the chromosome look like? So let's say that the chromatin has now condensed into chromosomes. So a duplicated chromosome is actually two chromatids. Oh, Herder, this is so many words. All right, let me give you an example. All right, here's one chromosome, okay? Humans, right, we have 23 pair of these. So let's just say this is your, this is your number 3 chromosome, let's say, right? from 1 to 23. Let's say this is your number 3. So before we make egg or sperm, or before we're going to just make any new cells in our body through mitosis, these cells have to duplicate. All right, so there's one of the chromosomes from the number 3 pair. What will happen is during the S phase of the cell cycle, which we'll get to in a moment, those chromosomes duplicate. Okay, so we had here one of the number 3. Well, that duplicates in S phase. And now notice we have 1, 2 sister chromatids. So this is now still a chromosome, but notice it has 2 chromatids because it duplicated in the S phase. These are identical. This is identical to that. These chromatids are identical. All right. Then, when we get to mitosis, which we'll go over in great detail here in a moment, those chromatids actually split and will end up going into separate cells. So this preserves our DNA, right? We had a number 3, right? And by the way, remember you've got 2 number 3s, so you might have another one over here that's doing the same sort of thing, but we're kind of focused on, let's say this is 3A, this is 3B, because remember humans, we have 2 of each. So each of these has to duplicate in S phase. So just to show you a little heads up of what's going to come up, right? These chromosomes are going to duplicate, they'll undergo mitosis, and then we'll end up with 2 cells from the 1. Okay? All right, so let's go over this cycle. How does this work? So here's the life of the cell cycle. So if you think about it, I like to think of it as, okay, we as humans or animals, we have a life cycle, right? We start young, we grow, then eventually we start to get older and pass. Well, cells have a very similar cycle, right? Most of their life, they're just kind of growing, hanging out like we are. There's just a very small part, the mitotic phase, where they're actually reproducing. So let's look at what these are. So the phases of a dividing cell's life. Interphase is the most populated phase of the cell cycle. This spends the most time, right? So let's divide interphase. And by the way, if you're looking at this chart, all of this, with the exception of mitosis, so not mitosis, but G1, S, and G2, that's all interphase. So in interphase, that's subdivided into these 3 parts. G1, think G, grow. The cell's just growing. Then we go into S phase. S phase, think synthesis. That's when our DNA is doubling or synthesizing, replicating our chromosomes, getting ready for division. Then G2, more growth. We're getting new organelles made, new biomolecules, because what's coming up? Oh, mitosis. That's when the cell's going to divide. So all of this is really about getting the cell ready for division. Then you see this G sub 0. Occasionally, some cells in our body go into G sub 0, what we call a non-dividing phase. There are a few cells in our body, some neurons, some liver cells, that once they reach their maturity, they no longer divide. So they go into this sort of resting state, or this G sub 0 state. All right, so after all of interphase, then we get ready for the mitotic phase. Mitosis is just the separation of the cell and the chromosomes. Specifically, mitosis is the chromosome separation, and something called cytokinesis is the actual cell separation. So, as you see here, cytokinesis is the cell dividing, the cytoplasm and organelles. Mitosis is really the chromosomes dividing. So where does cytokinesis fall? It kind of happens right here at the end of mitosis. So I'm just going to write that in there, cytokinesis. All right, so let's specifically get into interphase. So interphase, as I mentioned, is 90% of the cell life cycle. It's just doing its everyday job. Okay, so the cell cycle, all of this is interphase, as I mentioned, right? Not mitosis. And what's the cell look like during interphase? Okay, well, here it is. So basically, we see a well-defined nucleus, right? There's our nucleus in the cell. And you see this DNA, it's in the form of chromatin. It's not a chromosome yet. So interphase, think inter, between. So interphase is really what's happening between the phases of mitosis. So this is what interphase looks like. If you're looking at a microscope slide of interphase, if you see a nucleus and you don't see specific chromosomes, then we're probably in interphase. Most of the time, our cells are in interphase. Well, then mitosis. This is also called M phase, right? Mitosis, M phase. What's happening here? Well, this is all about copying the DNA so it can divide into the two new cells. So mitosis has 4 phases. So if we now take this section, mitosis, these are the 4 phases: pro, meta, ana, tele. And occasionally we'll see one in between pro and meta called pro-metaphase. You'll see that sometimes, not always. The way I remember this is interphase comes first, and if you go prophase, pro-meta, you might think of something, like something silly like your dog might say if it, had an accident. It might say, I pee-peed. Oops, that's supposed to be a P. I pee-peed on the mat. That's my way of remembering. Interphase, prophase, pro-metaphase, metaphase, anaphase, telophase. That's the order of the steps of mitosis, okay? So now we're going to go through and see what's happening in each one of these phases. All right, so we start in interphase, right? That's pretty clear. We see the nucleus, we see chromatin. Now, as soon as mitosis starts, right, there was the nucleus. Oh, also, we want to mention, we know there's an intact nucleus because we see this nuclear envelope surrounding the nucleus. And the chromatin, there's no chromosomes yet. Now we've moved into prophase. That's the first one. So prophase is characterized by actual chromosomes, right? Chromosomes, not chromatin. You see the chromosomes. And we start to see the nuclear envelope break down. Okay, notice the chromosomes and notice these centrosomes. These are an important part of mitosis. They're what's going to help guide the chromosomes and move them down these spindle fibers. So you'll notice that these centrosomes are starting to move to the poles of the cell. Okay, now we're in metaphase. What's happened? Okay, well, how do we get from here to here? Obviously, these centrosomes moved to the ends of the poles, and we now see these spindle fibers, these little green spindle fibers. I like to think of this as sort of like a little railroad track that kind of helps these chromosomes move. Well, where have they moved? To the middle. This is an easy one for me to remember because meta, they're in the middle. Well then, now that they've lined in the middle, notice that they split right down the center. The sister chromatids have split. So half of them go this direction to that centrosome. Half of them go to this direction. So this is anaphase. They've actually split from the center. We're starting to see new cells form. These are the new daughter chromosomes that are going to be in the new cell that is created. So telophase, notice that they've started to move. They're now closer apart. And notice we're starting to get, it's in animal cells, we're starting to get a little cleavage furrow. It's starting to cleave in and form this furrow. So that's telophase. And then finally, cytokinesis, which is not technically part of mitosis, but it's actually the division of the cell. Notice that the cell completely divided down the center, and now I have 2 cells ready to go through the process again. Okay? So you just need to think logically about what happens. We're in interphase, chromosomes condense into prophase. Then they move to the middle. Why do they move to the middle? Because they're going to split. They split in anaphase. They move to the opposite ends of the poles in telophase. The cell starts to pinch in to create 2 new cells, as we see happening as a result of cytokinesis. Okay, so now I'm going to show you some actual pictures, some photos of this happening. Just to remind you, all right, so here's prophase. We don't see any particular chromosomes. This is just the very beginning. You can kind of look really carefully and see them there. Maybe a little hard to make out. Notice then Metaphase. These chromosomes, these blue things, are moving towards the middle. Well, then in Metaphase, notice all of the chromosomes are right here in the center. What's the green stuff? Remember, that's the spindle. Okay? So we've got the chromosomes in the middle. We've got the spindle fibers that are kind of reaching out here. Anaphase, notice they've split. Notice the green fibers have kind of split as well. And notice that the specific chromosomes are starting to move. Late telophase, notice the chromosomes are in their different parts of the cell. We're ready for cytokinesis. Okay? So that was just another visual representation of how you might see this. So now let's talk a little bit how this occurs in plant cells. So we just saw in animal cells, but now we want to look at how it looks in plant cells. So plant cells are different for one really important reason. Remember, plant cells have this cell wall. And this is more rigid, okay? Animal cells just have that membrane. It's a little easier to divide. In cell wall, something else happens. And let's look at what happens. So here's interphase, right? We see a nucleus. We see our chromatin. Now we see prophase. Notice that the spindle is starting to form, and we see actual chromosomes once again. And the nuclear envelope has started to break down. Then we move to metaphase. Meta, the chromosomes have moved to the middle. Anaphase. Chromosomes separate, start moving to the poles. Everything so far looks pretty much the same as in animal cells. Here's the difference. For cytokinesis to happen, instead of a cleavage furrow like we saw in animal cells, we actually start to see the development of a cell plate. The cell plate will eventually turn into the cell wall. So that's the big difference. We don't see a cleavage furrow in plant cells. We see a cell plate start to form, so that will form the new cell wall. So now I want you to see if you can identify some phases, okay? So I'm going to show you a picture. This is of an onion root tip where a lot of mitosis would be happening, right? If you think of a plant, where is it growing? A lot of times at the roots, they're growing to go down to find more water, more nutrients. So if we looked at the root, we'd see a lot of mitosis happening. Why? Because that's where the growth is. How do we get growth? We get growth through mitosis. So I want you to pause the video for a moment and see if you can identify some of the phases of some of these different cells, okay? Go ahead and pause. All right, so let me point a few out. To me, most of these, it looks like, are just an interphase. So I'm going to put a big I over a lot of these. A lot of these that you just see around are an interphase, but a few are different. What you can see here is that we actually have some that we're seeing the chromosomes, right? It's not just the chromatin with the nucleus. We actually now see some chromosomes. So to me, this one right here looks like we're in prophase. Okay. This one down here, I would say we're probably in pro-meta. So we're moving towards the middle. Now here, notice these have separated. So this is either telophase or really late anaphase. Notice they have moved to opposite ends of the poles. Okay? So now that I've helped you with that one, see if you can find those. All right? So pause me again, see if you can identify some of the phases. Okay, how did you do? Well, once again, most of these are interphase. Remember, 90% of the life cycle of the cell is an interphase. To me, there's one that really stands out. It's this one. Clearly, to me, those are right in the middle, so that's going to be metaphase. Now, looking around again, some of these are a little more tricky. I would say this one's probably in prophase because we're starting to see chromosomes development, right? So that's an arrow saying this one's pro. So that one, right, telophase is what I would think for that one. Okay? There's a few more you can identify if you'd like. All right, so let's talk about how this evolved to wrap this video up. So mitosis, like everything else, had to evolve. Well, I've already given you a hint of probably how this started to happen. So we think mitosis in eukaryotes, right, that's us, likely evolved from the binary fission that I showed you earlier in bacteria. Why do we think this? Well, remember binary fission, one bacteria duplicates their chromosomes, then divides into two cells. Now, bacteria had a single circular chromosome. We have many linear chromosomes. So we think our mitosis evolved more than likely from this bacterial binary fission. Because you start with one cell, you end with two, and basically it divides up the chromosomes. Okay? So hopefully in this video, you now understand a bit more of how our cells have their own life cycle, and then how our cells divide through the process of mitosis.